Hello again, and welcome back to Science 7. So today, we're going to continue our talk about genes, about DNA and RNA, all this fun stuff. Today, specifically though, we're going to talk about the genetic code. So what we know so far, well, we've almost mastered our central dogma of biology, right? So DNA forming RNA, and ultimately forming our proteins, right? So we talked about DNA, we talked about the structure, the function, we also talked about DNA replication. And from DNA, we talked about this step here, forming RNA. And what's that step again? Well, it's transcription. So now we have RNA, and again, we've talked about the structure, the function of RNA. We've also talked about the process in which RNA ultimately goes on to form proteins. And that, again, is translation. What we will learn today is we will learn how to derive our polypeptide chain, right? So we're going to learn how to derive a chain of amino acids, right? So a polypeptide chain is simply a chain of amino acids. So before we get into that, let's go over a quick review. So with DNA, we have two strands, right? We have two strands. This is our DNA. One strand reading 5' to 3', and the other reading 3' to 5', right? So normally these strands are intertwined into a double helical form, and they run anti-parallel to one another, hence why one is 5' to 3' and one is 3' to 5'. And what's holding these DNA strands together? Well, they're base pairs. They're base pairs, right? So the four base pairs we have with DNA is A and T, A binding to T, and also G and C. Now these base pairs bind together through hydrogen bonds, right? We've also talked about how DNA is replicated, first of all, but then we also talked about how it's transcribed to form our RNA molecule, right? So our RNA molecule is going to be an mRNA. And our mRNA is also having base pairs, but however, these base pairs are slightly different than our DNA base pairs, right? Remember this. A, instead of binding to T, instead of binding to thymine, it's going to bind to uracil, or U. G and C, well, they still bind together, right? So this is nothing new. This is just a review. And then finally, our mRNA molecule is going to go on and aid to form our proteins, right? Or to form our amino acid chains. So this step, again, something we talked about before, is translation. Right, so in translation, how did we get mRNA to form our proteins? Well, if we remember, we had something called codons, right? So our mRNA had something to do with codons, if we can remember. And now codons are 3 base pairs in length. So let's say this is our mRNA molecule, this entire thing, mRNA. These are just its codons. So 3 base pairs in length could be anything. A, U, G, G, C, A, U, U, right? They could be any base pairs. But each triplet, each codon, codes for an amino acid, right? Remember we talked about this? So our codons code for amino acids. Today, we're going to look at how to build our amino acid chain. So let's say we had a chain of base pairs, such as this. such as this. This is our chain of base pairs. So the first thing we want to do is we want to determine if we have a DNA or if we have an mRNA strand, right? Because if we have a DNA strand, at first we're going to have to undergo translation to form our mRNA, and then we're going to have to go, transcription to form our mRNA, and then we're going to have to go under translation to form our codons and our base pairs, so forth. So how can we tell if we have a DNA or an mRNA molecule? Well, what's our difference? T base pairs is for DNA, and U is for RNA. So here, clearly we have T. So this is our DNA molecule. Now DNA does not form proteins, DNA forms RNA. So let's write the corresponding mRNA strand down. A is going to bind with U. T, A, that's a C, G, U, C, G, A, and A. So if we had any issues with this, we have to go back in our previous notes or previous lectures and learn how to match up base pairs. So now we have our mRNA molecule. Now we can go ahead and form our proteins, our polypeptide chain. So this is one of the ways we can do so, by looking at this chart here. So if we remember, it's codons that code for our amino acids. So every three base pairs is a codon. So in this case, we have 3 different codons. All we do now is we read off of our chart. So our first base pair is a U. So we look at our U row. Our second one is an A. So we look down our A column. And, because this is our second base pair, and our third base pair is a C. So we look for C. UAC, well, that's tyrosine. That's tyrosine. And we continue. G, so now we're down here. U, so we're in the first row here. And C, well, that's valine. So right now we have tyrosine, valine, and finally we have GAA. So again, G, our second base, is A, and our third is A again. Now we have glutamate. That's it. We just formed our polypeptide chain, starting with our DNA molecule. So this chart, or a chart very similar, will always be given to you on a test. You're not expected to remember what UCG codes for, right? So you'll always have some sort of chart. You just need to know how to read off the chart and come up with your polypeptide chain. This is another type of chart. Use this one if it's easier for you to comprehend. But let's say we had, let's try another one. Let's say we had something like U, A, G, G, U, C, C, G, A. Okay, so the first thing we're going to do is we're going to determine if the chain we have is a DNA molecule or an RNA molecule. Well, since we have uracil instead of thymine, we know that it's an RNA molecule. Next, we break apart our codons. So every 3 base pairs is a codon. And now we simply just read off our chart. So our first base is a U, uracil. So we start in the middle of this chart, uracil. Next, we have A. And finally, we have G. Now, what does this dark circle represent? That's not an amino acid, is it? Well, no, that's actually a stop codon. So remember we talked about stop and start codons? Well, here we have a stop codon. So technically, if we were actually synthesizing our polypeptide chain, we would stop here. But for our case, we're going to continue. Next, we have GUC. We have G, so we read from the center, G, and we go out, U and C, valine, valine. And finally, we have CGA, starting in the center, CGA. And we just simply write down our amino acid. So we just synthesized a polypeptide chain. I mean, we started with a stop codon, which doesn't make practical sense, but in theory, we now know how to read and synthesize a polypeptide chain.